drug discovery is a very tedious and time-consuming process. In drug discovery, we have a very important stage which is called lead identification. Now this lead identification, if it is done at alone, at single level with single gene or protein sequence, it will become very huge time dependent. To remove this problem, what we have at our hand is basically use of high throughput screening algorithms. So as we have already seen, high throughput screening basically is a silicon or in silico based method. In silico based method means where we have a laboratory setting in which we use silicon based machines such as our computing devices or robots for working on thousands of compounds in a one go. So similar to this high throughput screening, we have another mechanism which is called virtual screening. इस मॉड्यूल में हम ये देखेंगे कि वर्चुअल स्क्रीनिंग क्या है, व्हाट इज इट्स एडवांटेज एंड हाउ इट इज कंपैटिबल विद HTS or high throughput screening. So if I want to define virtual screening, what it would be? It is basically again number one in silico method. So it is done in laboratories. Then instead of using physical material or equipment, we have very huge or bigger sizes of databases which have information of already developed proteins or already discovered proteins or macromolecules. Now what we do whenever we get to know about a new discovered protein or macromolecule, we just compare this macromolecule with already stored macromolecules in our virtual library or virtual screen databases. So basically what is the definition? Basically what we see whenever we have new chemical compounds, we compare them from large database with the help of computers to find out subsets or candidate compounds for laboratory testing or drug discovery mechanisms. So virtual screen, what is virtual screening then? It is basically creation of large in silico virtual library of compounds. This virtual library means that it is not physically based. It is a record based thing which is in the form of electronic media using computational devices. On the other hand, what is high throughput screening? So as we already said that high throughput screening can be defined at two levels. One is at physical level and the other one is virtual screening level, which is at the software level. So high throughput screening gives us options of performing experiments in on thousands of proteins or gene expressions in one go. So what are the other different advantages? So there are different advantages such as we can have lead identification of thousands of proteins in one go. Then we have inclusion of candidate drug molecules. So inclusion means that when we perform our experiments on the given proteins, if they are relevant or if they are druggable are those molecules which can be converted to make some successful drugs. If they are okay in that sense, then we keep them. Otherwise there is option of exclusion of that proteins or those undruggable molecules. Moving on, what are the other benefits of this high throughput screening or virtual screening? The biggest advantage, as I said earlier, is that it can compare thousands of compounds in one go. So thousands of compounds are compared with the unseen protein, and if there is some match, then unseen protein is considered to be useful, or if it is not there, then we can say that this protein is unseen and it can be added into our library for future work. Fast rate development. So if we have database, bigger or larger database of our already discovered proteins, then the new compounds can be developed at a very faster pace. Optimized lead selection. So since it is comparing unknown sequences with the already known sequences, so it will be having a optimal solution for drug discovery. Then confidence in candidates. If we have some common scenarios or functionalities between unknown sequences and known sequences, then we are more confident that our developed drugs are more useful as compared to unknown sequences. It reduces our time and cost effective, of course, because it is already stored in our bigger databases. So it will have a direct effect on cost and our efforts with respect to time and money. So what are different virtual screening techniques available? So for virtual screening techniques available, we have shown here two techniques. Number one is molecular docking and the other one is similarity based virtual screening. Now what is molecular screening? In molecular screening based approaches, number one is molecular docking basically. So molecular docking, what we usually do is that we have a list of known sequences and then we have an unknown sequence. What we try to do, we try to dock unknown sequence with known sequences. So if that unknown sequence is docked or merged with some known sequence, we say that this screening mechanism is successful and we annotate that sequence. On the other hand, in similarity based virtual screening approach, there is no information or no previous knowledge available about the given protein. So it is just similarity based approach which can be done using multiple sequence alignment or phylogenetic based approaches, but we don't have a previous knowledge about the given or unknown sequence.